Hi everybody. Today we are going to focus on the metabolism as our concept. We will look at the three main groups of food that we consume, which are your carbohydrates, fats, and proteins, and how they get metabolized and when they are actually utilized as a source of energy. Now, to start with, what does the word metabolism actually means? Metabolism implies a chemical reaction that takes place. It is broken down to two categories, and I'll talk about them in a few minutes. Those are catabolism and anabolism. But basically, chemical reactions that are going to help you create ATP and also maintain processes such as homeostasis and regular cell functions like cell division, cell growth, replacement of the dead cells, producing secretions, contraction and propagations of electrical signals, contractions of muscles, and basically anything in your body that requires energy. Again, when we're looking at metabolism, there are two main categories or breakdown for metabolism. We have seen this earlier in the semester. We have talked about catabolism, which is basically breaking down your large organic molecules to smaller components and eventually synthesize them to create your ATP and or other high energy compounds such as your NADH or FADH2s, which are again, these are high energy electron transport molecules. The second type of metabolism we have is called anabolism. These are when you take smaller components or smaller building blocks and utilize them to synthesize something larger or more complex. So if you take a look at the picture we have here, here is the three main groups of food that you have. Again, triglyceride is the most common type of fat we have in our body. Glycogen is identified as a complex carbohydrate, and proteins are basically any type of protein you consume coming from either the vegetables or your animal sources. So when you break down triglyceride, you will get fatty acid and glycerol. When you break down glycogen to the complex carbohydrates, you typically get simple sugar, and the best example for that would be glucose. And by breaking down your proteins, you will receive amino acids. So if you look at these gray arrows you have here, notice that these gray arrows are going both directions. So if you're going from top to bottom, that's the process of catabolism, which is again breaking large molecules to smaller things and eventually utilize them for production of energy for high energy electron carriers. Notice if the arrows are moving upward, so you're moving from the second column up, basically taking fatty acid or glycerol and producing triglycerides, taking glucose and combining it to get glycogen, or taking amino acid to make proteins. That's the second type of, or second process within metabolism, which is known as anabolism. Taking something simpler and making something more complex, or synthesizing something more complex. Now, by looking at this picture, one of the things that I want you guys to notice is that the organelle that is going to be involved in the process of catabolism, as is depicted here, is a mitochondria. So mitochondria is going to be a focus of our lecture as we go through this process. It kind of highlights some of the essential importance of essential components, again, as we go through this. But to just kind of start you there, giving you the idea, to produce ATP, which is again our goal in terms of energy molecule or high energy molecules, we have a process called TCA cycle, also known as the Krebs cycle, and we have a step called electron transport chain. Now, notice that in these processes, you'll also produce byproducts such as CO2, water, and of course ATP, which is the actual product or our goal. You also consume oxygen during this process, which makes this an oxidative process, or oxidative phosphorylation, as we will learn later on. Again, this is kind of highlighting the same process that I've discussed with you guys, but in a little bit more detail. So here is my three main food groups, again, proteins, carbohydrates, and fats. If you take the most common source of energy for our body, which is carbohydrates, it gets broken down to glucose, which is the simplest and most effective type of sugar your body uses, but you also can use other type of sugars. Now again, to eventually use carbohydrates and produce ATP all the way at the bottom, what I require is a process called glycolysis. Glycolysis starts with the glucose molecule, and through this process, you form something called pyruvic acid or pyruvate. Pyruvic acid eventually converts to what we identify as acetyl-CoA, and acetyl-CoA enters the cycle we identified as CTA or Krebs cycle. Krebs cycle eventually transfers these molecules to the last process, which is oxidative phosphorylation, which is part of the electron transport chain. And as a product, the final product of all of these processes we're describing, you are producing ATP. Again, highlighting a couple points for you here. A couple side products is the waste gas CO2 that we produce. We produce water, and this process is known again as oxidative phosphorylation, which implies that you require oxygen input to be able to produce ATP. Now, the other part that I want to point out is even though this is described in terms of carbohydrates, you notice that there are arrows coming from both proteins and fats. So if we catabolize your protein, you produce amino acids, and amino acids can enter these processes at different locations. Again, they can be introduced as part of the process within glycolysis, which leads to formation of pyruvic acid. They can come in as acetyl-CoA, or they can come in as a product within the Krebs cycle. Same thing with glycerol and fatty acids. They can introduce within these processes that we're describing during the process of catabolism, and all of them eventually lead to formation of ATP. Now, kind of looking at the overview of metabolism, and again, bringing everything we're describing here together. Metabolism is important in terms of growth, production and secretion of material. If we have extra, it is stored as reserves for later utilizations of your body. It is used for maintaining your homeostasis and repairing any damage to your body. If you don't need something, it's usually formed some molecules that are complex through the process of anabolism. And if you require those, they break down to smaller molecules through the process of catabolism. The main organelle involved in this process is your mitochondria. Notice that when you basically utilize mitochondria, 60% of energy, quote unquote, is actually releases as heat, and 40% is what actually converts to ATP and is effectively utilized. One last thing to note here is this heat that we are describing in terms of metabolism is a big contributor to your core body temperature. So if you guys recall, your body temperature is maintained under homeostasis at about 98.6 or 37 degrees centigrade. And for your body to be able to do that, you rely on your metabolism to contribute a significant amount of heat for that maintenance. Now again, because carbohydrates or sugar is going to be the most common type of or source of nutrients for our body, we're going to start by describing what happens to a glucose molecule as it goes through all of these processes to end up with producing about 36 ATP as a final product. So you start with your glucose molecule. So here is one single glucose molecule that is described as C6H12O6. So you have 6 carbon, 12 hydrogen, and 6 oxygen. You utilize about 6 oxygen molecule, and again, 6O2, it's not an atom, so 6O2, the molecule. And again, the byproduct of this process would be 6 CO2, which is your gas, 6 water molecule, which contributes to the water your body requires, and 36 ATP. So from one glucose molecule, you're producing 36 ATP. Now, one thing that you do need to notice is the ATP doesn't produce or starts as ATP. It actually needs to go through electron transport chain, and it needs to utilize electron carriers or high energy electron carriers to be able to effectively accomplish that task. So as we go through these processes, you get familiar or you hear these two high energy electron carriers multiple times. One is NADH and the other one is FADH. Again, they're referred to as high energy electron carriers, and they are going to be extremely important for the process of electron transport chain, which produces majority of the ATP for your molecule. Now, let's start with each of these processes. So as we described, we have glycolysis. We have production of what we consider acetyl-CoA, which is not listed here because, again, it's not a full step. We have what we identify as Krebs cycle or citric acid cycle. And the final process is electron transport chain, which is, again, uses oxygen to produce ATP. So let's start with glycolysis. The first thing I want you guys to know is the location of glycolysis. Glycolysis takes place within your cytoplasm, which is basically the space within the cell. Glycolysis, for it to start, it requires ATP to initiate the process. So you utilize 2 ATPs. And at the end of the product, you produce 4 ATPs. So your net product of ATP is 2 ATPs. You also create 2 high energy molecules called NADH. So you have 2 NADH at the end of this process. So I'm going to draw this for you guys to make it a little bit easier to follow. So if we're looking at the process of glycolysis, go back 1 second. So if I'm looking at the process of glycolysis, what do I need to know? So glycolysis is you're starting with a glucose molecule, okay? And you are ending up with a molecule called the pyruvate or pyruvic acid. Sorry, sometimes my pen doesn't write very well, as you may notice. So when going from glucose to pyruvate, what do I need to do? First of all, I need to give this process 2 ATP. So basically, think about you don't make money unless you put some money in first. So here's your 2 ATP coming in. Now, as this process happens, you actually get 4 ATP from this process. So basically, your net ATP production is 2 ATP, right? You give back your 2 that you used, so you end up with only 2 left. You're also going to bring in 2 NAD+, which are basically your high energy electron carriers. And again, these won't make sense until the end of the process. So just kind of keep track of them. And you are going to have 2 NADH coming out. Again, if I ask you where does this take place, it takes place as part of the cytoplasm, which is the space inside the cell. So if here's my cell, here is my nucleus, right? The space, this is your cytoplasm, right? And that's where your glycolysis takes place. Now, again, keep track of these numbers. Right now we have 4 ATP produced, but the net production is 2 ATP and then 2 NADH molecules. Now, the next step that we have is we need to bring in the 2-pyruvic acid that we made into the mitochondria. Now, again, before I continue with that, I want to introduce one thing to you. And this is showing you what the mitochondria actually looks like. So if I draw a mitochondria by itself, it's a double membrane, if you guys recall that from earlier lectures. So here is the double membrane. And imagine I'm zooming into this. So here is one membrane, here's my second membrane, and this is inside of the mitochondria. So if I'm showing you kind of a zoom in member, this is the outer membrane, right? This one and this one are the outer membrane. This is the inner membrane, and here is the inside, which is the space here. So if this is my space inside, I'm gonna write M for that. I can't write the whole word. That's the matrix. So this is the space inside the mitochondria. This is my inner membrane. This is my outer membrane. And the space between these two is called intermembrane space. Now, if you're looking at this space right here, this is the space within the cell. That's your cytoplasm here. And basically what you have created up to this point, you have your pyruvate or pyruvic acid. So what I'm going to do is I'm going to transfer this pyruvate through this double membrane, the outer membrane, the intermembrane space, and the inner membrane space, inner membrane of the mitochondria, and bring it into the cell. And while I'm doing this, while this process is happening, I am burning about 2 ATP to accomplish this task. So imagine you're transporting something, basically you're gonna use some energy. Again, this comes into fact when you're doing your count and how you get that 36 ATP that we discussed from one glucose molecule. That's where that comes from. So now I'm bringing my pyruvate in, and I'm going to convert that to acetyl-CoA. So I'm going to take the pyruvate that I brought in, or again, pyruvic acid, and it's going to be converted to acetyl-CoA. And as this conversion is happening, what I'm actually doing is I am getting rid of one carbon in a form of CO2, and there's 2 of them, so I'm going to do 2 CO2s, and then I'm also producing 2 NADH2, or NADH, sorry. Now, you're wondering what the heck is happening here. So let me backtrack for you guys and show you a quick thing. So if you're starting and going back to what we start as the original molecule, a glucose is identified as a 6-carbon molecule. And that should be very easy for you to remember, since I started by describing glucose as C6H12O6, which implies that there's 6 carbon. So how does those 6 carbon look like? 1, 2, 3, 4, 5, and I'm going to put 1 here. Okay, so 6 carbon. Again, not the actual shape, but we're trying to understand the concept. So when you go from glucose to pyruvate through the process of glycolysis, you're actually producing 2 pyruvate molecules, and these pyruvate molecules are 3 carbon each. Is it going to be, and we're not looking at the chemical aspect of this. We're not looking at chemistry or biochemistry. We're trying to keep this as simple as possible right now, guys. So all you need to know is during the process of glycolysis, which is depicted up here, I started with a 6-carbon molecule, and each of these circles represent 1 carbon, quote unquote. By the time the glycolysis ends, I'm going to end up with 2 pyruvate, which is as to say, I've taken a 6 carbon and chopped it up to 3 carbon molecules, and I get 2 of these. Now I have transferred these 2 pyruvates, so you can put a 2 here. I have taken these 2 pyruvate, and through a process, I have converted these to 2 acetyl-CoA. Okay? Now again, the pyruvate are each what? 3 carbon, right? 3 carbons. Acetyl-CoA is only 2 carbon each. So here's my 2, here's my 2. So what happens to those extra carbons that we have? What happens to these 2, let's say, that were at the end? They were released as what? CO2. Now, again, as a byproduct, now I have also produced 2 NADH. So up to this point, I have used, up here, I have used 2 ATP, and I produced 4 ATPs. and I also produce 2-NADH. I have transferred the 2-pyruvate into my matrix of the mitochondria, and for that process, I use about 2 ATP. And then when take that 2-pyruvate, I have converted that to acetyl-CoA. This conversion has taken the 3 carbon molecules and converted them to 2 carbon molecules. So 3 carbon to 2 carbons. What happened to those carbons? They were released as CO2. And again, we also create 2 high energy molecules, which are your 2-NADH. So as of right now, I have my acetyl-CoA inside the matrix, and I'm ready to start my process of Krebs cycle. So how do I do Krebs cycle? So give me 1 second, guys. Let me skip this and I'll come back to it. So I can do one quick thing for you. And I just have some extra pages, blank pages, so I can do this for you. Okay, so let's talk about Krebs cycle. So Krebs cycle, which is the next process, is start inside the matrix. So I'm just going to show you this again. Here's my double membrane, right? So I'm not going to label them, but I'm hoping that you guys know this is the mitochondria basically. So that's the outer membrane, that's the inner membrane, that's the intermembrane space, and this space right here would be the matrix of the mitochondria. So I have brought in the pyruvate, right? And then eventually converted that pyruvate to 2 acetyl-CoAs, right? And each of these acetyl-CoAs are again 2 carbon molecules. Now we're talking about the Krebs cycle here, right? So Krebs cycle. So it's a cycle. So as this cycle is happening, I'm introducing the 2 acetyl-CoA into it. So 2 acetyl-CoA is coming in. And what am I actually bringing with these 2 acetyl-CoA? I'm adding a 4 carbon molecule from the previous cycle. Do you need to know what this 4 carbon molecule is? No. All you need to know is that it has 4 carbon attached to it. And now I am combining this 4 carbon molecule with a 2 carbon acetyl-CoA. So I'm going to end up with a 6 carbon molecule, right? So 4 carbon from the previous cycle, and you see that coming together as we finish this process. A 4 carbon molecule from the previous cycle came in and attached itself to the 2 acetyl-CoAs, 2 carbon acetyl-CoA, and as a result, now you have a 6 carbon molecule. Now, as this cycle continues to happen, the 6 carbon molecule becomes a 5 carbon molecule. Again, do you need to know the name for these molecules? No. You will learn it in microbiology. You may learn it if you take biochemistry, but for our purpose, we're focusing on biology, so names are not necessarily important for us. What I do want you guys to understand is if you are switching from a 6 carbon molecule to a 5 carbon molecule, anytime you lose carbon or your carbon is decreasing, that means it's coming out as CO2. So 6 carbon to 5 carbon, that's 1 carbon dioxide. But remember, this cycle is going to happen twice because I have 2 acetyl-CoAs. So you're actually producing 2 CO2 when I'm talking about 1 full glucose molecule. Now, what else do I produce? I also produce 2-NADH. Now, next step, 5 carbon molecule is going to go to a 4 carbon molecule. And you're like, wait, you already have a 4 carbon molecule here. These two are not the same molecules. So what do you do when you go from a 5 carbon to a 4 carbon? You are producing, again, 5 to 4. That means you're producing 2 CO2s. You're producing 2-NADH and you also produce 2 ATP. If you wonder where that ATP comes from, again, not our focus. You guys are welcome to go and review this in a biochemistry class. Now, you're going to go from a 4-carbon molecule to a different 4-carbon molecule. And as this transition is happening, now you'll produce 2 other NADH, and you also produce 2 FADH, FADH2s. Now notice we haven't heard FADH before. This is the first time we're hearing it, but that doesn't matter. It is still considered one of our high energy electron carriers, and it will be used during the process of electron transportation. So I want to kind of go over this, and you guys can just listen to me, or you can go back and review what you have. But I want to kind of reiterate some of the points. So we started with one single glucose molecule, which had 6 carbons. We went through the process of glycolysis, and during this process, I have utilized 2 ATP and produced 4 ATP and 2 NADH. I have taken, the final product of this process is 2 pyruvate molecules. Each of these pyruvate molecules contain 3 carbons. I then transported these 3 carbon pyruvate molecules into my mitochondrial matrix, passing it through the outer membrane and the inner membrane. And during this transition, I used about 2 ATP, so I have subtracted another 2 ATP. When I take the 2 pyruvate and convert it to acetyl-CoA, I go from a 3-carbon molecule of pyruvates to 2 carbon molecules of acetyl-CoA. And anytime I am losing a carbon, that comes out as CO2. Because I had 2 acetyl-CoAs, because I started with 2 pyruvate and produced 2 acetyl-CoA, then I have also released 2 CO2 molecules. I also have produced 2 NADH during this process. Now I'm going to take my acetyl-CoAs, which are 2 carbon molecules, and introduce them to the Krebs cycle, which is happening inside the mitochondrial matrix. The 2 carbon acetyl-CoA is now going to combine with a 4-carbon molecule from the Krebs cycle, and as a result produce a 6-carbon molecule. 6-carbon molecule, as it goes through the cycle, it loses one of its carbon and it becomes a 5-carbon molecule. And as the transition from a 6 to a 5-carbon molecule, you are releasing 2 CO2s as well as 2 NADH. You can go from 5-carbon molecule to a 4-carbon molecule. Again, because you're losing carbons, those come out as CO2s. You also produce 2 NADH and 2 ATPs. Lastly, you take your 4-carbon molecules and convert it to a different 4-carbon molecule. And during this transition, you also produce 2 NADH and 2 FADH2s. Please do note that going from a 4-carbon to a 4-carbon, I do not have any CO2 being produced because the CO2 is only produced when you are releasing carbon or decreasing your carbon number. And again, these 4 carbon molecules can now interact with the new acetyl-CoA from the product of the glycolysis and continue the cycle. We'll come back to our white pages. Here is the same thing that I depict for you guys. It's just a little bit more confusing than the drawing I have, but you're welcome to take a look at them and review it. Notice that it says the products is 8 NADH, 2 FADH2s, and then 2 ATPs. Now, if you look at the cycle I was showing you earlier, you have 2, 4, and 6 NADH. So why are they saying 8? The reason for that is they also consider the 2 NADH produced going from pyruvate to acetyl-CoA during this process. So you can count them separately, or you can count them as part of the Krebs cycle. That's up to you. So let's create this chart before we go to electron transport chain and take a look at what we have so far and what is that translates into terms of ATP. So we had our glycolysis process, right? We had our acetyl-CoA, and again, some people write this as part of the Krebs cycle. I don't like that, so I write it as a separate step because then you can keep track of them. Then you're going to talk about the number of NADH that you produced, the number of FADH2s, the number of ATPs, and then final ATP count, basically. And again, it will make sense as we go through this why I have a regular ATP and a final ATP count. So during glycolysis, I produce 2 NADH and then 2 ATP. Formation of acetyl-CoA also produced 2 NADH. Krebs cycle produced 6 NADH, 2 FADH2s, and 2 ATP. So keep this in mind. We'll come back to these numbers to get a final count of our ATP. A quick note on that, you also make a note, you utilize 2 ATP to transfer pyruvate into your matrix of the mitochondria. So keep that in mind. We'll come back to it later on. Now, everything we have done so far in terms of production of the NADH and FADH2s are in preparation of utilizing those in the electron transport chain to produce ATP. So I'm going to show you my drawing first, and then we'll take a look at the picture, which kind of highlights the same information. So again, looking at my mitochondria, we have the double membrane, the outer membrane, inner membrane, and the intermembrane space. This space is your matrix of the mitochondria. Now, within the inner membrane, you have 4 proteins. So I'm going to just draw them as boxes. I'm going to keep it simple as much as we can. So 4 boxes. And at the end of these boxes, I have this kind of, I guess a mushroom, I guess if you can call it. So these are protein, literally I'm going to call them protein 1, 2, 3, 4. And then this is my ATP synthase, as in it is responsible for production of ATP. Now, what I'm going to do is we're going to start with using my NADH. So let me change a different color and then we can walk through this. So let's just start with our NADH. So NADH comes in and drops off its electron and it becomes NAD plus. Okay, so NADH, let me erase this and then rewrite it. So you end up with NAD plus. So the electron that's being released in this process is going to be a high energy electron. So it jumps to the first protein. From the first protein, it jumps to the third protein. From the third protein to the fourth protein, and then eventually comes back down. So here's my electron coming back down. Now, as it's making its jumps from the first to the third to the fourth, it moves 1 hydrogen from this space, which is your matrix, into the intermembrane space. So here's 1 hydrogen moving up. Here's another hydrogen moving up. And here's my third hydrogen moving up. So now, as I'm moving this, and this is continuously happening, as I'm moving the hydrogen into the intermembrane space, which is the space here, there will be an increase in hydrogen concentration right here. And now we're gonna go back to the whole concept of diffusion. You have high concentration of hydrogen here, low concentration of hydrogen here. What does hydrogen want to do? Hydrogen wants to come back down and diffuse. But what is it going to diffuse through? What path can it take to diffuse back into the matrix? And for that, it goes through the ATP synthase. So as these hydrogens are flowing back in, going from a higher concentration to a lower concentration, it causes the ATP synthase to turn, okay? This turning, and again, we're not going to get technical, allows me to convert ADP, an inorganic phosphate, to ATP. So hydrogen moved up, right, into my intermembrane space. The concentration of hydrogen in the intermembrane space went up. So those hydrogens want to diffuse back into the matrix. The way they can do that is to go through ATP synthase. And as these hydrogen are flowing back in, it allows the ADP to attach a phosphate to itself and form ATP. So now I have hydrogen here, right? I have a hydrogen. Well, actually, you can take two hydrogen. You take two hydrogens, you take your electrons, and you take, here's the keyword, guys, half of an oxygen molecule. And what do you create? You produce H2O, which is water. So I have moved 3 hydrogen up, choosing my NADH. Every time 1 hydrogen comes down, I produce 1 ATP. Therefore, because I moved 3 hydrogen, I have produced 3 ATP as a result of this process. So 1 NADH was able to produce 3 ATP for me. Again, what is the byproduct of this process? Hydrogen. What is another name for production of ATP? It's called oxidative phosphorylation. Oxidative, right? Phosphorylation. What does that mean? Oxidative means you need oxygen. Phosphorylation means adding a phosphate to produce your ATP. So here's the most important aspect of this concept so far. If you lack oxygen, so if there is no oxygen here, hydrogen will not be accepted, which means the hydrogen concentration stays high here, which means what? There is no flow of hydrogen back in because high concentration is present here, which means ATP will not work and no ATP will be produced. So if you have no oxygen, there would be no oxidative phosphorylation, which means there would be no electron transport chain. Now, if I'm repeating the same process for our second high energy molecule, which is our FADH2, again, same thing. You have FADH2 going to FADH. And what is it releasing? It's releasing an electron. Here's the key difference between the two. FADH2 releases the electron into the second protein. That electron jumps into the third protein, jumps into the fourth protein, and again, the electrons will come back down exactly like we described. But FADH2 only moves hydrogen in number 3. So hydrogen is moved at number 3 as well as number 4. There is no hydrogen moving into the intermembrane space in this second protein. So same process is repeated here. Hydrogens come back down, ATP synthase work, ADP and phosphates are attached, you produce your ATP. Oxygen, the electron, and the hydrogens are combined together to form your water as a byproduct. The main difference between NADH and FADH2 in terms of their involvement and the ATP production is, one, NADH uses protein 1, 3, and 4, versus FADH2 uses proteins 2, 3, and 4. Second difference is NADH moves 3 hydrogens and therefore produces 3 ATPs, versus FADH2 only moves 2 hydrogen and therefore only produces 2 ATP. Okay, so hopefully that makes sense. Now that we have an idea about our oxidative phosphorylation that is happening in our electron transport chain, let's go back to our count here. And let's see if we can figure out the total number of ATP that's being produced. So if glycolysis contributed 2 NADH to our electron transport chain, every of those NADH produce 3 ATPs, so that adds up to 6, right? So 6. And then we also already produced 2 ATP. That gives us a product of, or final product of ATP from glycolysis to 8 ATPs. Acetyl-CoA produces 2 NADH, which each produce 3 ATPs in the electron transport chain. That gives us this number of 6. In Krebs cycle, which is really, really effective in production of high energy molecules, you have 6 times 3, right? You have 2 times 2, right? Because each of the FADH2s only produce 2 ATPs, and then 2 ATP. So that's 6 times 3 is 18. 2 times 2 is 4, right? So 18 and 4 is 22. And then 2 ATPs, that gives us 24. If you add these numbers together, you will get 38 ATP, right? But remember I said utilize 2 ATP to transfer pyruvate. So now we're gonna subtract those 2 ATPs. So what is our magic number we end up with? 36 ATPs, which is what I described for you at the beginning. Every time you use 1 glucose molecule, you're gonna end up with 36 ATPs. I know it's a lot of concept, guys, but you guys do need to review it to make sure you guys are comfortable with this. The numbers are important for me in the exam. You need to know what each of the steps make in terms of high energy electron carriers. You need to know them in terms of ATP production and how those ATPs and FADH and NADHs are add up during the electron transport chain in terms of the numbers of ATP they produce. Now here's a same thing that I draw for you. It's just, it seems more complex, but it's the same concept. So let's walk through this. Here is your mitochondria matrix. Here's your inner membrane, the outer membrane, and the space between that is your intermembrane space. Here are your 4 proteins, so Roman numerals 1, 2, 3, and 4. Here is your ATP synthase. And again, walking through this, your NADH drops its electron, and you move hydrogen, hydrogen, hydrogen. So 1, 2, 3 hydrogen moves. You can follow the jumping of the electron. So electron jumps from number 1 to 3 to 4 and then comes back down. As this hydrogen comes back down, ATP synthase are going to convert ADP and inorganic phosphate to ATP. You also will have hydrogens, electrons, and half of the oxygen combined to form your H2O. For FADH2s, the electrons jump from protein number 2 to number 3 to 4 and then come back down. And during this process, you're going to move hydrogen in FADH, in protein 3 and 4. Again, notice number 2 has no hydrogen movement. And when those hydrogen comes back down again, you produce the byproducts of water. So electron transport chain requires oxygen. Oxygen is the final electron acceptor, as we described, to form the water. If you do not have the electron carrier, if you don't have basically the electron acceptor, not the carrier, honey, electron acceptor, then that would be basically stopping the whole electron transport chain. So that's the scenarios where you will have limited oxygen in your body. And for that, we'll talk about another process, which is utilizing fermentation as a mechanism of production of ATPs. But please do keep in mind that when we're talking about electron transport chain, it's extremely important for you to have oxygen as the electron acceptors. Otherwise, majority of the ATP that's being produced in your body, that production will be halted, and therefore you will be unable to basically produce about 80% of the ATP for your body utilization. Okay, I'm going to stop here and we'll continue this PowerPoint by looking at the process of fermentation, by looking at how you break down fat and proteins, and also different stages of starvation. So keep tuning.